All right, hello everyone and welcome to our 4th online cell modeling workshop organized by the Center for Reproducible Biomedical Modeling. My name is Veronica Perevsky and I'm excited to introduce today's workshop on data aggregation with my co-organizers, Sam and Yosef. So getting into the motivation, which you guys may have seen before, using mathematics and computation really help us to model biological systems in order to be able to test hypotheses, design new systems or adapt existing ones, and even predict behavior of biological systems, which we otherwise wouldn't be able to do using only experimental methods alone. So as a community, we're really working through challenges that still exist in our modeling workflow. And this workshop series is designed to bring together experts who can present on very specific topics and their new developments in modeling software and tools that would be able to address some of these challenges and also to provide suggestions for the broader challenges in the field. So today's workshop will address the challenge of data aggregation. We also want to invite you to participate in our upcoming session on numerical simulation of biological models in October and to view our previous workshops online on our archive. So to go through the agenda really quickly, I'll provide a short introduction on data aggregation and then we'll move on to two 20-minute tutorials of data aggregation platforms and conclude with a group discussion. Our goals for the workshop series are really to be able to identify common challenges we all face in our everyday modeling workflow, share our experiences with data aggregation, brainstorm new solutions, and continue to foster this community we have of modelers. So during the discussion, all comments are welcome. We want this workshop to be useful for you, so please give us any feedback that you do have. And we would appreciate if you could mute your microphones during the tutorials and during the discussion when you're not asking questions, just so that we don't get any background noise. So today's challenge is to be able to collect the data that we need to construct and validate our models. This includes data that's used to assign parameter values, perform parameter estimation, inform simulation conditions, and validate that our model predicts known biology using experimental measurements. Discovering data that's appropriate for our particular biological context, for example, the species we're studying, the cell type, disease condition, or some intracellular pathway of interest, can be difficult and time-consuming, especially considering the growing collection of open-source data available online. And even though we do have this increase in data sets that are available to everyone in the modeling community, that data that is available is often still incomplete as far as our specific unique models are concerned. So it's unlikely that we're going to be able to obtain all of our measurements for our model solely from the data that's available or without some alteration. So we may need to approximate measurements using similar values or related molecules or related organisms, or we may need to estimate a value from a distribution of possible measurements. This is especially difficult, you can imagine, for very large-scale models like models of the entire cell, or even models of an entire organism where the whole cell model requires many different types of measurements to inform different pathways, and the model of the human would require information about specific cell types, specific organs or tissues that are used to inform the model. Currently, data repositories are widely distributed, making it difficult to ensure that our search for data is the most relevant to the model and is returning the best results. So to ensure that the massive amounts of data that we are accessing for our modeling process is going to be able to allow us to collect the best data possible, we really need tools that can effectively automate the data collection processes using constraints that we can provide as experts on the topic. And we need tools that can ensure that we can perform exhaustive searches of relevant measurements in available data sets. So the solution to these challenges come with the discussion of data aggregation, which involves the collection of published measurements tailored to a model's particular biological context from literature and data repositories in order to integrate them into our model. Biological data aggregation platforms are currently being developed to automatically access relevant data in response to user searches and make it accessible for integration into biological models. There are currently many biological databases available, and these may provide specific types of data that we need for a modeling process, such as a particular type of measurement like RNA or protein expression data, or data with diverse measurement types, all related to a particular disease or a particular species of interest. The primary challenge comes with efficiently accessing the most relevant data for our particular model. So data aggregation platforms that exist today work to access the data available within existing databases and literature, and to perform some validation on the data to ensure it can be converted to a consistent description format, and make the data accessible to the modeler through indexing and specific search terms. So these aggregation platforms that we have may also manage metadata or and even add annotations that unambiguously describe the data sets. So in order to get a better idea of how to tangibly use some of these platforms, we want to welcome our invited speakers for our workshop today. So today we have Dr. Yasset Perez-Riverol and Yosef Roth. And our first speaker for today will be Dr. Perez-Riverol. He is the proteomics team coordinator at the European Bioinformatics Institute, and he'll present on the Omics Discovery Index, or OmicsDI, which is an open source platform that's used to discover and share omics datasets. So welcome, Dr. Perez-Riverol.
Thanks. Do I need to share my screen, I guess, yeah?
Yeah.
Okay, let me share. Okay, share. Can you see that? Okay, cool. Thanks for the invitation. My name is Yasset Perez-Riverol. I am the team coordinator of proteomics service at MBL-EVI, and also the project coordinator of Omics Discovery Index. Finding a publication is really easy. I mean, anyone can find a publication going to EuroPMC or PubMed. If you want to find a specific topic, let's say about proteomics or about genomics, you don't need to go anymore to Google. You can go to those services and then find a specific publication. However, finding the data set is not that easy. And this is mainly because there is nothing out there, or there were nothing out there to be able to find a data set. And this happened to be a time-consuming process that go through going to a specific databases or going to consortium databases, like for example, Metabolome Exchange or Proteome Exchange, and try to find out the data set, or GEO, for example. At the end, you end up by going to PubMed and then try to throw the publication, trying to find the specific data set. And sometimes you will end up in Google. Then what OmicsDI tried to do at the very beginning was to find out a way to aggregate multiple resources, multiple archives that contain omics data, and try to provide also functionalities for searching or recommending data set across all these databases. And the ultimate goal would be to produce also tools that enable to provide to the user what actually are the more interesting data set for them, okay? And we come up with this a specific, let's say a specific pipeline that try to, for every partner of omics, OmicsDI, let's say a proteomics database or a genomics database, we translate that into a common representation, and this would be the OmicsDI XML file format. Translate that into a database, which is a Mongo database. And then on top of that, we put a lot of pipelines for the automatic annotation, manual annotation of data, and finally indexing all this data to being able to search. We started in 2017 with around 8 partners. So far by 2019, we have the major databases in the 3 omics field, like metabolomics, proteomics, and the 4 omics field, metabolomics, proteomics, genomics, and transcriptomics. But we also have a lot of data from the model field with all the biomodels data set. And these are the statistics around in 2019, how much data do we have. When the user, if you see also with OmicsDI, because all the data is in the same place, What I mean when I mean data, I mean the metadata of the data sets. You can see some trends about how the data publication is coming right now. And for example, you can see that proteomics and genomics and transcriptomics are still the more the fields that produce more data. But for example, proteomics has been growing so far in the last recent years, and also metabolomics. The second thing is, with the metadata, we also can share specific information around the tissues, of course, the taxonomy of the organisms, but also the cell type and all the instrument that was used to generate the experiment. OmicsDI do not only aggregate the data, the metadata coming from all these resources, but also try to find links within the data set. Because one of the interesting things that we want to achieve is that, imagine that you are a proteomics researcher or a genomic researcher, but you don't know what happened at the proteomic level, let's say. Then you know that in EGA or ENA, you can find some, you have some data of transcriptomic data in your lab, but you don't have proteomics facility. And you want to see out there what kind of proteomics data is close to your genomics data set in terms of the same taxonomy, same disease, or same speed, or same tissue, but also which data sets are out there that actually are multi-omics data sets, okay, which are really relevant for the development of models and things like that. Which is, imagine you want to find all the studies that the proteomics, metabolomics, and transcriptomic data has been performed. And this is not an easy task. If you want to see, for example, a data set and a study that contains the 3 omics types, you will need to go to each specific omics database and then get the data set from there. With OmicsDI, we can link all the data set from for multiple omics that belongs to the same study. Okay, how do we do that? Well, we first actually know from the publication where a data set actually, a metabolomics data set, a transcriptomic data set actually contains some proteomics data. And this is a really nice example when you have a multi-omics data set, where actually this is you are actually seeing here the transcriptomic data set, but you can see here, you are actually see here the proteomics data set, but you can see all the related data set, the metabolomics data set, the transcriptomic data set related with it. Then in OmicsDI, wherever you find this icon here, this is a multi-omics data set, and you can actually search for those data sets if you want to see multiple omics for the specific type of a study, okay? That's the first thing you can do. The second thing is OmicsDI provide, I would say we want, at the very beginning, we will call ourselves like the Amazon of omics, because we want to provide functionalities for powerful search functionalities. For example, in this case, you can actually search all the data set where this gene actually has been found expressed or differentially expressed. For example, in breast cancer, then you will have all the proteomics or genomics data set where this gene is relevant for the data set. This is not only, what I want to highlight here is that the search capabilities of OmicsDI is not only at the metadata level, but also at the biological entities. All of our partners actually release also with the genomic, with the metadata, also which are the more important biology entities related with the data set. Let's say you have a proteomics data set, there would be the protein that has been identified in this particular proteomic experiment. If it's a transcriptomic experiment, all the transcripts that actually change the expression, genes that change the expression in the transcriptomic experiment. And these are released to OmicsDI, and OmicsDI can use that to provide better search capabilities, okay? Then you're searching here with the metadata, but also the biological entities. Then we want to allow to suggest to the user interesting data set. We have implemented a suggestion system and recommendation system based on 3 metrics. The first one is the publication. Imagine you are in a proteomics data set and you want to see all the other omics types that are related by a publication, the same publication. Then if you are looking to a proteomics data set, you can see all the omics type, transcriptomics, metabolomics, and so on, that are within the same publication related by the PubMed ID. The second thing you can do is actually doing by the biological entities. And that would be, imagine you are in a proteomics data set, we can compare the biological entities. That means if you are in a proteomics data set, we can compare to all the other data sets in proteomics that share the same molecules. You have the same list of proteins that are relevant to 2 data sets. We can recommend that to you also. And the other thing is about metadata. We can recommend to you all the data sets that are related to you, take into account only the metadata, specific keywords and so on. For example, this is a data set, transcriptomic data set. And then here we have all the list of similar data sets close to this data set, taking into account only the metadata, okay? And this one is the second way of recommending data sets, is basically based on biological entities. This is a specific data set where all these data sets share a huge amount of proteins with the same list of protein of the data set that you're looking at. You can define a threshold. I want to see all the data set that share with me, let's say, in some sort of way, 50% of the list of proteins, okay? And the last thing is related with reanalysis, which is basically, we can trace all the data set that has been reanalyzed in another database, okay? Then this is a really nice concept on, basically we can say which data set has been reanalyzed by another data set. This concept in the biomodel field would be, biological biomodels would be, if you have a model that is based in another model, then this is considered a reanalysis. Is you have some previous knowledge that actually is used to generate another type of model or a new knowledge. In the proteomics or transcriptomic field, imagine you have used some part of the data set to reanalyze this data and obtain new claims or new way of analyzing the data. There is different patterns for reanalysis, and the main pattern we see for proteomics and transcriptomics is the one data set is reanalyzed multiple times. However, for example, in the biomodel field, we have seen, and this is really easy to detect, where one model has been reused multiple times over the years, okay? And then multiple models are based on a specific part of this model. And you can build more complex networks when one model is actually coming from multiple models. Why this is important? We want to give value to some specific data set. This is a recent publication we published in this year in Nature Communication, because the amount of data we have so far is really huge. We want to actually say what kind of data cannot disappear from the public, but also what kind of data is more relevant to your specific queries. And for that, we have different metrics. Reanalysis is one of them, citations, number of views, downloads, and so far and so forth. And we have provided this specific score you can see here when we collect all these metrics for each data set. I am skipping a couple of these slides, but what you can actually see, for example, is you can go to OmicsDI, is www, sorry, we should say this, I should say this at the very beginning, www.omicsdi.org. You can go there, and now you can say your queries, for example, proteomics, and then you can sort all the data sets that are out there in proteomics You can sort by the number of reanalysis. The number of reanalysis, how many times this data set has been used by other groups or other databases to obtain new claims. And in case of a biomodel would be how many models are based on this original model. And this gives you an idea how important is this model or this transcriptomic data set or this proteomics data set for the community. And this is not only by the reanalysis, also by the number of citations. For example, we go into EuroPMC and PubMed and try to detect how many times a model or a proteomics data set or a transcriptomic data set has been cited directly into a publication. It's not a citation to the original paper, but the citation of the original data set. And with that, we can see how many times a data set has been reused also in different publications, okay? I will not go into detail here because it's a little bit of more data. But this is the way we detect the number of citations. For example, you can see here, this is a transcriptomic data set from GEO, and you can see here directly the citation to the GEO accession. Then how much, I think the publication, you can go to the publication, read the whole publication, but one of the interesting points we want to make is how much each community is reanalyzing data, how much each community is reusing data. And for example, the transcriptomic community, you can see here that it's actually reusing data sets that are 10 years old. And this is really relevant because other communities, when a data set in proteomics is published, within 3-4 years is not used anymore, okay? But when it's a transcriptomic data set or a genomics data set, then the number of reuses still keep growing within time. And this talk about, for example, how mature is, for example, the technology, how, for example, how you take decisions around how to reuse a particular data set that is in a particular field that is actually within 3 years or 4 years time, then probably you don't want to use it because it's too old for the time being. And we try to establish some sort of correlation between all these metrics. We have found that there is no correlation within all these metrics, and that means that all of them should be used to try to detect what is actually relevant to you. Then when you search in OmicsDI, you should go and then see, for example, if you're, go back to my previous example, if I search for breast cancer in proteomics data set, then you should look for the number of reanalysis, the number of views, or the number of downloads for the specific data set. The last feature we have implemented in OmicsDI, and I think this is actually really important, is we want that researchers deposit more their data. And for doing that, I think we need to give some credit to whoever has produced the data and make their data public. Then in OmicsDI right now, you can actually log into the system with ORCID, and you can claim your data sets. You can actually say, oh, these are all the data that I have produced. And you can see that each data set has a score with considering all the metrics that I have explained, reanalysis, number of downloads, the number of views, citations. That means that you can have some sort of Google score around the data sets. And that will encourage, or we hope this will encourage users or the community to put more data out there, but also to put, for example, in their grants or their CVs, the OmicsDI profile, where people can actually say, oh, I have put outside, I don't know, these 20 papers, and this is my Google H index, but also this is, for example, my how much data I have put outside from OmicsDI, and you can check in OmicsDI and how much my data has been reused by the community. It's actually interesting here. OmicsDI has been around for 1.5 years, but actually is within the 60% of traffics of of the resources on EVI traffic, I would say, and this is really relevant. This means that a lot of people is using it to search data across multiple omics, and it's actually becoming a well-established resource that you can use to search data. With that, I am almost done. I want to thank to all the partners of OmicsDI. As I said before, we have metabolomics data, proteomics data, transcriptomic data, models, and genomics data. And if you have your database or you want to include your database into OmicsDI, you just need to send an email and we will guide you through the process of adding in your database. Also, if you have queries about how to search data in OmicsDI, it's really simple to contact the team and the team will help you. Thank you.
Awesome. Thank you so much. That was a really excellent tutorial. Before we go on to the discussion, we're going to have our second tutorial, and then we can discuss both of the platforms together. Okay. Do you mind? Can you stop your screen share? Thank you. All right. Sorry. All right. So now we're going to welcome Yosef. Yosef is a PhD candidate in the Carr Lab at the Icahn School of Medicine at Mount Sinai, and also one of my co-coordinators for our workshop series. So today, Yosef will present Datanator, which is a software tool that can discover, aggregate, and integrate various types of data needed to model whole cells. So welcome, Yosef.
Thank you, Veronica. Thank you everyone for coming. It's nice to be on the presenting end of this workshop. And thank you, Dr. Riverol, for your Perez-Riverol for your presentation. So I'm going to present a Datanator. So first, I guess a description of what it is. It's I guess in comparison to OmicsDI. So OmicsDI is like an integration aggregation of data sets, and it's designed to help you find the most relevant data set and its connection to other available data sets. A Datanator is, I guess, solving a slightly different problem. It's organized by the data itself. So it's a software tool designed to help one find the most relevant data for modeling or for a given biological parameter. So first, I'm going to present some background on what my lab researches and why we in particular were motivated to create such a tool.
Hey, Yosef, can I interrupt you for just a second?
Yeah.
So could you possibly change your slides? So right now they're presenting in presenter view, so the...
oh, you can see, yeah, that's pretty funny. Thanks, Abby. How do I do this? No. This is better, right?
Yep, that's great.
Thank you. Okay. Okay, quick background on what my lab is researching. So my lab studies whole cell models, and the goal is to create a mechanistic model of every process in the cell. So when creating these models, we're interested in questions like what types of data are required to construct a whole cell model. So it ends up being a whole array of different types of data, like, for example, DNA sequences, RNA abundances, protein abundances, metabolite concentrations, reaction kinetics, many different types of data. And this data is, I guess, distinct in two ways. One, the data varies by type, and it also varies by the data repository. As in, if one would want to go to public repositories and collect this data, they're often located in different repositories. So to give a motivating example, let's say one was going to model the metabolism using Cayless-Menten kinetics. So let's say we just wanted to model this one rate. We're going to end up with 3 different data types. And if someone were to collect this, they would have to go to 3 different data repositories. So one thing that would be necessary is metabolite concentrations. And one location where one might get this is the UCMDB database, a database of E. coli metabolite concentrations. Another data type that would be necessary is reaction kinetics. And one example of this is SAVI-RK. And last, someone would need enzyme concentrations. And one location to get this data would be PAX-CP. So even to model with just one rate, we need 3 different data types. And the location of this data is found across 3 different repositories. And this aggregation poses a challenge to the modeling community. Our lab 2 years ago actually had a survey where we surveyed different researchers in the modeling community. And one of the questions we asked is, what are the most time-consuming aspects of your research? And the most popular answer at 52% was data aggregation. So any tools that our community can make to accelerate this process will greatly accelerate the field in general and can enable much more large-scale models than are currently possible now. So I'm going to outline various challenges on data aggregation and then describe how they are solved in a Datanator. So challenge number 1 is that the data is often distributed, right? You'll have to go to many different online repositories to get the data that you're looking for. So we solve this in Datanator by unifying all the data to a single repository. We do this in Datanator's database. Another challenge, the data is typically sparse. Let's say you're looking for ATP concentration in E. coli or something like that. Oftentimes you'll find that there isn't any data on that you can find. So what you want and how we solve this is we create tools to find similar data, right? Maybe there's no ATP concentration data in E. coli, so you can find ATP concentration in Bacillus subtilis or some related organism. Another challenge is that the data is typically noisy. Often there won't just be a single value, right, for a biological parameter. There'll be a whole distribution of experimental data. And this poses a challenge for modeling if you want some single quantitative value to help construct the model. So we solve this by creating tools to reduce the data, some representation of it that can be aided, that can aid in model construction. Another challenge is that aggregation is very tedious. It takes a long time to aggregate the data and then to integrate it and unify all the definitions. The way that we solve this is we automate all the possible steps in data aggregation. And I should just point out that Datanator, it doesn't fully automate the data collection process. Some aspects of it still require manual oversight and decision making. But the goal of Datanator really is just automate everything that can be automated and accelerate as much as possible. And one last challenge is that it's difficult to record the data that's used to construct a model. I mean, maybe some biological parameter is informed by a whole bunch of experimental data, but it's difficult to actually record the data that went into a given parameter in a model. And the way that Datanator solves this is by connecting the models more directly to the underlying experimental data so that someone trying to understand the model can see how it's generated, and it's much more reproducible. So the way Datanator achieves this is we have an integrated database on the back end, and users can interact with it currently with our website, datanator.info. We're also working on a Python interface as well. Okay, so now to give a bit of background about the Datanator's integrated database. We've collected many public repositories online. Here's some examples: PAX-CP, SAVI-RK, and added them to integrated database. And we also added in some prediction tools like kinetomic string comparison. This can predict chemical similarity of compounds, and Ezyme, which is a reaction similarity tool. In addition to integrating all the data onto a single database, we've unified the biological definitions and units, standardized it all. In instances where the data only exists in its raw form, we've processed it. One example of this is RNA-seq data. Right? One location this is RayExpress, but it's stored mostly in FASTQ files. So we've processed it to transcripts per million unit, which is more usable in a model. This is the current state of the database. We have data on DNA, RNA abundance, protein abundances, taxonomy, interactions, rates, metabolite concentrations. This is from like a whole array of different repositories online. We're continually expanding our work and hope to continue integrating various databases. So now, just to go back to our motivating example to describe what tool that we would want. Let's say we're trying to model a rate using K-less-Menten kinetics, we'd want a tool that does as follows. You can input a given biological parameter, maybe the concentration of ATP, along with model specifications, say Bacillus subtilis in certain experimental conditions. And we want this tool to output a consensus value. It's something that can be used to help construct the model, as well as the whole distribution of data that was used to generate the consensus value. This is the functionality Datanator provides. So this is the data collection process that one would use in Datanator. And it's all drawing from our integrated database that integrates data from all the public repositories. So step one is to identify the data that you're looking for, or the distribution of data. You want to specify the biological parameter, maybe concentration, along with the biological conditions like experimental conditions, the species, environment, etc. Okay, once a distribution of data has been identified and filtered to find the most relevant data set, each one of the observations should be reviewed to just ensure that it's accurate and credible, seems like it's right for the model. Once that has been finalized, step number 3 is to reduce the data to a consensus value that can then be used to help construct a model. And last, this should be recorded, right? The consensus value or representation of data, along with the total distribution, should be recorded and saved. So now I'm going to go through each of these 4 steps in a bit more detail. Okay, so step 1 is to identify the most relevant data set out of all the data that is in the database. And the user will want to use a series of filters to help find the most relevant data set. So one filter you might want to use is chemical similarity. So let's give an example. Let's say a user is looking for ATP concentration, maybe in E. coli, but maybe no data exists. The user can use Datanator string comparison to find data on chemically similar compounds. So maybe ATP, AMP, ADP, these are chemically similar, and maybe he can use concentration data from there if nothing is available on ATP. Another potential filter is biological similarity. A user will want to find the most taxonomically relevant data that exists. So ideally, this would be for the target organism that's being searched for. But if no data exists on the organism you're looking for, the user would want to filter to find the most taxonomically related data available. And a third filter is environmental similarity. A user is going to want to try to search for experiments that took place in biological conditions that mirror what's being modeled or studied. After the most relevant data has been identified, the user is going to want to review each one of the experimental observations to ensure that it's credible and should be used in the final distribution. Once the distribution has been finalized, the user will want to reduce this to a consensus value that can be used to aid model construction. And last, the total distribution of data along with the consensus should be recorded. So just to give a quick summary before I'll show you how this can all be done. The 4 steps are 1, to identify the data and filter to find the most relevant subset of the total data. 2, to review, to look through each one of the observations in the relevant set to ensure its accuracy. 3, to reduce this distribution to a consensus value. And 4, to record everything, the distribution and the consensus. So now I'm going to show you how this can be done. So everyone, if you want, go to datanator.info, Datanator, DATANATOR.info. Okay, so right now the front end can support searching for metabolite concentrations and protein concentrations. We're continually expanding the front end to include other data types. The next one that we're going to include is reaction kinetics. So let's start with metabolite concentrations. So let's say a user was searching for ATP concentration in E. coli. Okay, so the first thing is we see the total data in Datanator. We have a series of filters we can apply to find the most relevant subset. So one is taxonomic distance. Right now, it's including all the data on all cellular organisms, but we might want to limit it to just E. coli, which would exclude the other data, which was yeast. So, maybe we'll limit it there. You can also limit it by media type. Maybe we just want Gutnick media as an example. It's up to the modeler to decide what seems most relevant. Then we can look through, each one of these experimental entries to review the data. Maybe we think, oh, this medium contains acetate. That's not representative of experimental conditions, for example. All right. Then once the data has been reviewed, we can click Get Consensus, and that'll reduce the value, reduce the total data to a representation, a model construction. It'll also show you how the filter data compares to the total data before filtering. And you can keep updating this. Maybe you want to include all the data. You see how that affects the consensus value. Good. Now, sometimes there isn't actually data available for the metabolite you're looking for. So let's say we're looking for UTP. Maybe we want it in yeast. Okay, so we see that there isn't actually any data on UTP concentration in yeast, just in E. coli. So maybe a user will think, all right, well, E. coli is too distant from yeast for this data to be of much use. So we can click on Include Similar Compounds. And what this will do is it will use kinetomal strength comparisons to include data on structurally similar metabolites. So we see now we actually have a whole list of other metabolites besides UTP. And this can be filtered through this molecular similarity filter. This corresponds to the kinetomal comparison. So we can put it all the way to 1, which is just UTP, or we can allow other types of molecules. So let's say we wanted to filter and just include things, metabolites from yeast. So we see we actually have a number of structurally similar metabolites to UTP. And the user might decide that these are actually relevant to inform UTP concentration. The user might want to put the filters the other way and insist on a very specific molecule or with a general taxonomy. And now it's UTP, but it's in E. coli. And that'll give a different consensus value. Both of these tools are available, and the modeler can try both filters in parallel with one another. We try to provide, I guess, the most tools to the modeler to make these sorts of decisions. Now, another type of search a user would want to do is for protein concentration. So protein searching in Datanator is actually organized by the KEGG orthology number. So KEGG orthology is a general orthology of proteins. And every KO number corresponds to a specific protein, but abstracted from a specific genome. So let's give an example. Let's say we were looking for a phosphofructokinase. So there are a number of KO numbers. But let's say we took this KO name, 6-phosphofructokinase 1. So this KO number corresponds to that protein, and there are many different UniProt IDs. And this is because UniProt ID corresponds to a protein in a genome. And every genome will have a new UniProt ID for what we would consider to be the same protein. And this is true even within substrains of bacteria. The same protein in E. coli will have a different UniProt ID between different strains. But it's important for Datanator's purposes that there's a way to reference proteins that's abstracted from any specific genome. For example, one of the things we were able to do with searching for metabolites was we were able to find data on the most closely related organisms. So if there's no ATP data, maybe in Bacillus, we could look at ATP data in E. coli. So similarly with proteins, it's important to have a way to reference the same protein, but across different organisms. So that's why it's arranged by the orthology number. So let's say we're looking for phosphofructokinase. Then we click on the KO number, and we see all the different UniProt IDs and organisms that has abundance data for this protein. So one, we could do get consensus. We can look at the distribution of the total data. But of course, if we're looking for an organism, we can make it more specific. So maybe we'll look at E. coli. So then we can update our consensus and look at the protein concentration data that way. Once once we've looked at gotten the consensus value, we can actually save this data. This will be a text file that includes the total data, the filter data, and the consensus values. And this can be saved along with the model. And then it will actually document how the experimental data gave rise to the consensus values. So just to give a quick summary of the data collection workflow. So Datanator allows you to identify the most relevant data using a series of filters. 2, you can review each one of the experimental observations to ensure its accuracy. 3, it allows you to reduce the data to a consensus value. And 4, it allows you to automatically record total distribution of data, the filter distribution, and the consensus values. So for our future work, well, 1, we hope to incorporate a whole bunch of new databases. There are many public repositories, and we hope to continue integrating them into Datanator to allow more data to be searched. We hope that this will become a communal project. This is a tool that the modeling community needs to progress, and it's beneficial to many people. So we hope that many people will be able to contribute to integrating individual data repositories into Datanator. This can be a communal project. So thank you to the Car Lab. Especially thank you to Yang, Saad, and Balash, and Bilal, who've worked the most with me on developing Datanator. So thank you. And thank you, Veronica, for moderating.
Thank you, Yosef, for a great tutorial. Now that we've seen both of our tutorials for today and have some practical uses for the 2 data aggregation platforms, we can move on to our group discussion. My co-organizer and I have prepared some questions, but please feel free to jump in if you have questions. And I guess I'll first open it up to anybody that might have questions right off the bat about either of the 2 aggregation platforms presented. Okay, I guess one question I am curious about would be whether you could both comment on what you think the main advantages to Datanator or OmicsDI in comparison with other data aggregation platforms.
Well, I think for OmicsDI would be that there is nothing out there yet to aggregate data from multiple omics types in terms of the original data, the archival data. I mean, there is a lot of things in system biology, let's say, where you can try to aggregate specific experiments, but there is nothing like PubMed for omics data types, which basically, and I think this is the biggest strength of or feature of OmicsDI that allow users to search any type of omics data that is out there and that is public. And I will say this is the main advantages of the resource. Yes. Yeah.
And for Datanator, I think similar to the advantages of OmicsDI, but I guess on the data level, there are many types of functionality, like filtering, that can only really be done if the data itself is integrated. For example, like if one would want to compare at the same time chemical similarity, environmental similarity, and taxonomic similarity, this can only be done if the data is all integrated and filtering tools exist. So there are many excellent data repositories online, and I guess they all host their own data in a very organized way, but they don't have the same sorts of functionality and integration that one would need to quickly get the data necessary to construct a model. So Datanator allows one to do that relatively quickly.
Okay, we can move into some broader questions. So in terms of trying to determine whether data is relevant to the model you're building and ensuring that the quality of the data is sufficient to inform your model, how is the metadata actually evaluated, I guess, on the back end to ensure that a data set does meet the search specifications? And also, how is the metadata stored and then distributed when data is downloaded by users or integrated into their model?
I think, well, for OmicsDI, the metadata is stored in the same way in the same way that was provided by the repository. We have seen that different omics types has different level or detail level of the metadata. For example, transcriptomic and metabolomics actually as a field, they actually provide quite a lot of information around the sample metadata at the level of even the individuals. However, for example, for proteomics, that's not the case. And sometimes you only have some minimal metadata around the experiment, but not that much around the sample metadata, and which is the most important part to be able to reuse the data. OmicsDI actually do not force you to provide more metadata. It is kind of a hierarchical level when we have data sets that do not have enough metadata, I would say, for the proteomics in this case, but also we have data sets that are really well described. And we don't touch that. We just provide that to the community, and whenever you search for the metadata or with keywords, you will find the things that are described in a better way, I would say. I think OmicsDI is trying to promote right now with the proteomics community more descriptions around the data sets. And I would say also we are trying to standardize in OmicsDI the three communities, proteomics, metabolomics, and transcriptomic, in order to also have kind of the same metadata file format, serialization file format, that will enable eventually the user to download the same file to describe whatever type of omics data. And that's what we are working towards that, yeah.
Yeah, I mean, on our end, for one thing, because I guess we've integrated different data sets, it's sort of like a puzzle, but oftentimes you can import metadata from other places. So like one example is if like the UniProt ID fits into a KEGG orthology, you can get a lot of additional information about a protein from what KEGG has in their database. And if you can link up, gene names, you can get more data that way. I think this is sort of common in lots of, many of these different omics databases where you have to link one database up with everything else. But what we do, I guess, on our end is we do unify all the definitions. So it ends up being pretty standardized, at least on our database. And I guess we try our best to let users search in as many ways as possible. So like a user, certain things have like database IDs, like UniProt ID or KEBI ID for metabolites. Things also have names, so we try to accommodate all the different ways it might be searched.
So it sounds like at least, or it kind of sounds like for both platforms, there's not necessarily data that would be excluded based on quality. Is that accurate? Like essentially you make data available based on the metadata you have and its relevance to users, but don't necessarily perform additional checks that it meets some like sort of minimum quality standards.
I think in OmicsDI we have some standards to include the database, and that's okay. On a data set. You're right. I mean, we work with the provider, and we don't include things that, for example, will disappear eventually in a year time. We don't include things that do not have any, for example, title or a species or things like that. But as you said, as I said before, we have different level of metadata. And if the provider actually has a minimum metadata, which so far is an identifier, a title, a description of the data set. And I think taxonomies are like organisms that, and I think that's it. Of course, the omics type, then it would be, you will be able to be included. We don't have any checks on, you know, the quality of the metadata, or we haven't thought about that, you know, like saying we will rescore the things, take into account the amount of metadata that you provide. We don't have that.
It ends up being difficult because for most things, there isn't really a standardized metadata. Like one example is like a growth media, or you know, other growth conditions. There's just really no standardized way to represent this data. So oftentimes metadata will be there, but just in like a non-structured way. So it'll be difficult to even make such a criteria.
Okay, I guess it would be nice to hear if either of you would identify major challenges in the field of data aggregation. Like from your perspective, what is holding us back?
From my point of view, the major thing that probably holding back most, I mean, really interesting multi-omics experiment, especially. I mean, I think if you try to use a transcriptomic data set, you will be able to reuse it. Or if you are trying to use a proteomics data set, you will be able to reuse it. But I think the near future, will be able to reuse actually multi-omics data sets. You know, if you have some transcriptomic data and you have your model, for example, and you want to use some proteomics data that is out there, I think this is when things get more complex. And I think the first thing we need to solve is the same way, we should describe the same way in the sample data. And that's what we will try to work toward with providers in order to be able to describe in the same way the transcriptomic, proteomics, or metabolomics experiments with the same the experimental design in the same way. Same words, same ontology, same, hopefully same file formats. Yeah? And that's the first thing. And the second thing I would say is we need to work more with the provider to being able to put more data out there, especially the relevant data. I have the feeling that in some fields like metabolomics and proteomics, still the more biological interesting data is not out there and it's not easy to retrieve. And even if we move forward with OmicsDI, and that's why we give priority to the impact and claiming by the authors and researchers need to be able to get some credit about the data that they have been deposited. I mean, this is pretty clear in publication. If you have a publication, you put out there in PubMed, you will get a Google Scholar, you get your index and people know who you are, and you get your grants around your publication. It's not the same about data. People need to get some credits and get some rewards about putting the data out there. And that's what OmicsDI has been working so far the last year. And the last thing I would say is about how do we report the biological claim? And that would be the ultimate aim. If you report, for example, the proteomics ID or a protein, we should be able to report a protein with an accession and for example, an expression value that can be correlated with the transcriptomic, that can be correlated with the gene. And that would be need in the future more strong reference system, for example, to translate a gene ID into a transcript ID, into a protein ID, and so forth and so forth. And I think that's the main challenge for the future. Yeah.
Thanks. Yeah, I very much agree with Dr. Perez's answers. I think one of the major challenges is just the inconsistency of the way things are described. For many different types of data, I guess take RNA-seq for example, there's just incredibly more data than anyone has time to actually go through. And theoretically, if you're looking at a certain biological process, there'd be tons of data available. One of the major challenges is that because it's not described in a consistent way, someone has to go through the metadata of each one and decide whether it's relevant for what's being studied. Like there's no automated way to just look up, data from this mutation or from like normal, wild type conditions. So there needs to be some sort of standards for how samples are represented to actually allow people to take advantage of the enormous data that's already available. The second problem I think that Dr. Perez mentioned is that the majority, I think, I don't actually know the majority, many different data on different, I guess, for different data types aren't going into like a public repositories. So they're described in a publication, but it's not, I guess, automatically accessible by anybody. And someone would have to actually go through the Excel files or something to get the data, and that doesn't really scale. And yeah, there need to be incentives for people to actually add them to repositories. Also, groups like Savvy RK actually go through the publications and then add it to their own repository. That's also like a major benefit and helps overcome this problem.
Thank you both for giving some of your challenges. I want to direct it to the audience and just check if anybody else has questions they would like to ask either of our presenters.
I'll ask a question. So in light of the very large amount of omics data which has been made available publicly, but which is not adequately annotated with its metadata to be useful, do you think it would be worthwhile for a funding agency to support a project to make as much of this data as possible cost effectively better annotated so it could be useful?
I think if you go to, for example, in my opinion, is like EBI right now, MBL EBI is working right now in the problem of trying to standardize the way we submit data to multiple archive, multiple omics type. I think, as I said before, if you go to the transcriptomic field or genomic field, independently fields, you will be able to find out and the level of annotation is well enough, I would say, for the standards that we need. Yeah, probably not for automatic reprocessing. Probably is not enough. And I'm sure someone can come up with a really nice grant around that. I think OmicsDI actually was developed within the BD2K grant like 2 years ago in US. And actually, this grant actually put quite a lot of efforts into develop systems to annotate and cross-link different data set and omics type. And I think someone can come up with a really nice grant to annotate, especially relevant data sets, yeah? But the problem that I think will demand more work, and I think grants should be, I think there is a lot of potential for public funding for doing that, is actually when arrive to multi-omics types, yeah? When actually your community, the model community is really relevant for that because you're a consumer of multiple omics, yeah? Then I think in that sense, there is a lot of work to be done. There is a lot of work to be done in a sense that if you said, I have a multi-omics experiment when I have proteomics, genomics, transcriptomics, I want the same sample described in the same way. There is nothing out there that enables you to do that. There is not even tools that interpret the data from those annotations. EBI is now running, has been going for 3 years, and it will be running for even more. A project to standardize the way we submit data to MBL EBI resources like EGA, ArrayExpress, and PRI, and metabolites. But this has been going for 3 years, and I think it will be going for more, and it's a core resource is core funded. But I am really sure if you or a community came with a grant, and you can write me an email if you want, and we can actually try to be involved. I think there is a lot of potential to write grants or ideas around standardizing the way we write or annotate metadata, and we exchange data across multiple omics. And that's I think, yes, yeah, there is a lot of potential for that.
I can say one other use case that I guess I know of where better metadata would have been helpful. So certainly if you're comparing omics across fields, but I think another instance is, I guess, really large-scale analyses of the data itself. So I know Avi Mayan's lab, I think part of the LINCS consortium, they were doing like an analysis on all the data and all the human data in ArrayExpress to find correlations between genes. And one of the things that they did was they were able to look at specific cell lines and I guess make the correlations more precise. But the reason they're able to do that is because cell lines is one of the, I guess, types of metadata that's actually well standardized in ArrayExpress. But there are many other specifications one might want to look at if there was a way to do it. So I think even within one data type itself, at a certain scale of analyses, it actually does require a better description of the metadata.
All right, I want to give the audience a chance to ask one more question. Otherwise, we can wrap up. All right, well, thank you, Dr. Perez and Yosef, for joining and sharing your tools and data aggregation expertise with us. It's been a very insightful workshop, and I think we all are better equipped to go out into the modeling unknown and find our data, so thank you.
Thank you for the invitation.
Thank you, Dr. Perez.